Hello everyone, thank you for joining today. My name is Kebisha Vasukala from Dr. Sebastian Vega's lab here at Rowan University. Today I'm very excited to present our work on peptide responsive synthetic receptors for spatial gene expression in 3D materials. As cells communicate with their surroundings through receptor-ligand interactions, including in hydrogel environments, our goal is to design a receptor platform that detects custom peptides, triggers orthogonal and reversible responses, and functions within the 3D materials. This technology has strong potential for translation into medicine and industry. Talking about the market, the cell therapy market is projected to exceed $45 billion by 2030. Similarly, tissue regeneration is expected to surpass $60 billion by 2033, driven by organ repair, wound healing, and biomaterial innovation. Despite this promise, current receptor platforms face major limitations. Most synthetic receptors lack orthogonality, meaning they interfere with natural pathways. They also lack reversibility, so signals cannot be turned off once triggered. As a result, specificity is reduced and crosstalk complicates the design. To overcome these issues, we developed a new receptor approach. Our synthetic receptor EBDA, which stands for extracellular peptide ligand dimerization activator. In EBDA 1.0, ligand binding triggered a signaling cascade producing a detectable output, which is a proof of concept for EBDA version 2.0. This work has already been published in SCS Synthetic Biology in 2024. In EBDA 2.0, ligand binding control nuclear localization of transcription factors, enabling specific gene activation. This precision allows us to design signaling pathways that activate only when and where we want. For example, immune cells could release therapeutic cytokines only as needed, or stem cells could differentiate at the correct stage during the tissue repair. To achieve this level of control, we design EBDA with modular domains, making it programmable and versatile. EBDA is modular, programmable, and functional even in 3D environments. Recognition lies on customizable peptide tag pairs such as HA, 3G, and flag. Inside the cell, a signaling domain transmits the signal to the nucleus, while an activator domain switches on target gene transcription. We tested this in HEC cells engineered to express the receptor and embedded them in peptide functionalized hydrogels made via click chemistry. Upon encountering the peptide, the receptor activated, producing a clear fluorescent signal, which is confirmed by confocal microscopy, and the images are processed and quantified using an image analysis tool. Next, I will summarize the results achieved with this system. We have developed the design for a synthetic receptor system EBDA that is orthogonal, reversible, and compatible with 3D hydrogel environments. This platform is modular, allowing custom peptide ligands to drive specific gene activation with spatial and temporal precision. Potential applications include targeted cell-based therapies such as promoting tissue regeneration or controlled immunomodulation in disease settings. Our next steps are experimental validation in cells, which will establish functionality and therapeutic potential. To summarize, this work lays the foundation for programmable cell signaling with strong potential in regenerative medicine and tissue engineering. So before I conclude, I would like to acknowledge the people and organizations who made this work possible. I would like to thank Dr. Sebastian Vega for his guidance and support, and Dr. Matthias Rechtenwald for his contributions to this project. Also, this work is supported by the National Science Foundation. Thank you all for your attention.